So welcome everyone today on our seminar, and it's my pleasure to introduce Sofia Gabriel, and first time giving a talk here.
Yes.
Okay, so it's a pleasure to introduce you.
Sure.
Sofia is from University of Lisbon. She's evolutionary biologist. She did a PhD in Lisbon, and she is specialized in the phylogeography of rodents mainly. Speciation processes, chromosomal arrangements, and genetic diversity, and recently more leaning toward the ecotoxicology, impact of rodents and rodenticides on the biodiversity. And today she will present us her project and some results.
Yeah, I will.
Please.
Thank you, Vijek.
Yeah.
Well, good afternoon, everyone. It's a great pleasure to be here. It's been I have been having a long-lasting relationship with CIBIO, so it's a great pleasure to actually be here on this side of the stage. Of course, I want to thank Vijek for hosting this session, but I also want to thank Raquel Vasconcelos and Paulo Célio, who were actually the original responsibles for making sure that I would come to Porto and share some of my work with you guys. So I'm not sure if Raquel is online, but if she is, thank you. So as Zbigniew said, I am. She is. So as I wrote in my bio sketch, I am a born and raised rodent biologist. And over the recent years, I'm kind of slightly diverging, still focusing on rodent-related subjects, but looking at a somewhat different perspective and enlarging my interest into other species directly impacted by rodents, and in this case, rodenticides. So I'm going to talk to you about the toxic legacy that comes from the impact of exposure to anticoagulant rodenticides on the pest rodents themselves, so the target species, but also on the non-target wildlife that preys on them. So most of my career, I've been focusing on synanthropic rodents, so rodents that are associated with humans. And among them, we have house mice and rats. And as all of you certainly know, they have historically been considered pests due to their deleterious impacts on human dwellings. Oh, this is deformatted. I should have checked it first, sorry. On human dwellings, we all know the hazard that is having a mouse or a rat inside our homes and inside our infrastructure. Well, they're not called rodents for no reason. They need to chew on things, and that behavior alone, to wear the teeth because they have continuous growth, they need to chew on hard stuff, and that's why many times they are even causing fires and destructing infrastructure. They're obviously also a huge hazard for stored products and crops. They are, as we also all know, a public health problem because they are the reservoirs of numerous zoonotic agents, pathogenic agents, and also they are a hazard to biodiversity. And here is an image, a bit of a gore image, I'm sorry for that, but it really shows the impact of such a tiny species on an albatross chick where, oh, sorry, I can move this way. Okay, no, it's okay. Maybe it's even working better. So Gough Island, it's like a very remote island in the middle of the Atlantic, and there's like a population of huge house mice that feed on the albatross chicks, and this is obviously a biodiversity problem because the population is being compromised. So these pests, of course, are responsible for major economic global losses that have been actually quantified. There was a paper from Diane et al. In 2023 that reached a number of $3.6 billion of losses, only considering rodents, okay? Between 1930 and 2022, 87.5 millions between the 1980s and 2022. So just a 22 years period, and this huge amount of loss. So to fight this huge battle against rodent pests, the use of anticoagulant rodenticides has become the most widespread and globally used method over decades since their early development in the 1950s. So this was just a short introduction. Now I will present you the outline of what I'm going to talk about. So first I will show you what are anticoagulant rodenticides and how do they work, and then move on to rodenticide resistance, concept and consequences. I'm going to show you data that we have accumulated on bioaccumulation in target rodents, and then bioaccumulation/biomagnification in non-target wildlife. And to wrap up, just a small summary and a take-home message. So as many things in the history of biology, many compounds are found by accident, and this was also the case for anticoagulant rodenticides. So in the 1920s and 30s, North America, mostly Canada and the US, were struggling because farmers were noticing that cattle was dying with internal bleeding, and they had no idea why. So they were feeding sweet clover hay to the cattle, and if the winter was too moist, it would get moldy, and the fermentation process of the coumarin that exists on the coumarol that exists on the sweet clover would be transformed and have this anticoagulant effect on the cows. So, but it was only in 1940 that in the University of Wisconsin, they discovered the molecule responsible for that, so the coumarol, and realized that that was the anticoagulant agent that was causing the bleeding. So the way that this works is that the coumarin will inhibit the vitamin K epoxide reductase, so the enzyme that processes vitamin K and recycles vitamin K, which is absolutely essential for the blood clotting process and the coagulation. So these animals were not coagulating properly, and that's why they were dying of internal bleeding. Exactly. So the death was caused by this huge and systematic hemorrhage. So it was in the early 50s, so in 1954, that the synthetic version of this coumarin, warfarin, was introduced as a rodenticide. So it was pretty much effective when it was, you're finding it the bacon and cheese flavor, right? Yeah. So this biocide was so effective that it became widely spread throughout the world. It was even later on incorporated in human medicine, and today, if either you or any of your relatives have like heart problems or needing blood thinners, actually this is the exact same molecule that you will be given, obviously in very, very small dosages. So these compounds will act as vitamin K antagonists, blocking the vitamin K-dependent cascade in the production of the blood clotting factors, leading to the internal hemorrhage and eventually the death of the target rodents. So initially, these compounds, I'm sorry, you'll have to bear with me with a few big names, but it's not really important. I'll just resume them into GARs and SGARs, okay? So first generation anticoagulant rodenticides were the first ones to be developed. So warfarin, the original compound, cometetralyl, chlorofecinone, and difecinone that were a huge success. But resistance to these rodenticides soon emerged. So it was in 1958, so just 4 years after the introduction of warfarin, that the first paper, well, back in the day when publishing in Nature was as easy as this, okay? This is the publication, simply saying, it came out in 1960, that over a period of 2 months, so that's quoting, a heavy rat population on a farm in the west of Scotland was treated with 2 anticoagulant poisons, difecinone and warfarin. Although the treatments were properly conducted and the rats ate the poison bait freely, little impression was made on the size of the population. There was no evidence of recruitment from outside, and the conclusion drawn was that this population was more than normally resistant to these anticoagulants. So this led to the rise and the development of second generation anticoagulants, including bromadiolone, brodifacoum, difenacoum, difethialone, and flucomefene. So what were the characteristics of these? They were more toxic, they were more persistent, and they were more bioaccumulative. So the first generation needed multiple feedings for the animals to reach the lethal dosage. However, the second generation only needed a single feeding of the lethal dose to actually lead to the animal's death, so potent and toxic that they were. As you may know, there is a delay due to the neophobia of these animals to particularly rats, neophobia against, well, new things, new objects. They made sure that there was a gap, a delay between the ingestion and the initiation of symptoms. So the animals wouldn't realize that the bad that they were feeling was a result of the ingestion of the rodenticide. But, spoiler alert, emergence to these also emerged. So it was in 1995 that Greaves published the first concept of rodenticide resistance. And he said, or he defined it as a major loss of efficacy in practical conditions where the anticoagulant has been applied correctly, the loss of efficacy being due to the presence of a strain of rodent with a heritable and commensurately reduced sensitivity to the anticoagulant. So that was the paper. However, it was only in 2004 that 2 simultaneous papers, again in Nature. So for some reason, apparently this subject was a hot topic at the time. 2 independent papers from 2 independent research teams published in the same number, as you can see, Nature 427. So just different papers on the same issue. Identified the gene that was responsible for this trait, the resistance trait. And they realized that it was the VCORC1 gene that encoded for the target enzyme that was responsible for the recycling of vitamin K. So how does this resistance trait works? So there's the wild type of the gene that produces the normal enzyme, so the wild type enzyme that leads to a susceptible animal that will theoretically die if eating the lethal dose of rodenticide. But then a huge number of mutations have been described throughout the 3 exons that compose the gene. So you can see here, many of them. I won't, of course, bore you with too much information on this. What I wanted to show with this slide was the inhibition effect of the first and the second generation mutated versions of the gene producing the mutated enzyme. That when expressed in yeast microsomes, the level of in the y-axis we have the level of inhibition of the mutations resulting in the decrease in susceptibility. Okay, so the higher the bar, the more resistance the animal will show. So as you can see, there's a few mutations that are highly important in this resistance trait, and that's for FGARs, I'm sorry. And then many of them are the same. Most of them in exon 3, which is the most important one, which mutations are more impactful in the resistance, also show an inhibition effect on the enzyme. So we have here for bromadiolone, for difenicum, for diphtheolone, and for brodifacoum. Come on. No? Okay. So because the plot wasn't thick enough, in the house mouse, there was another layer to this story, which actually on the place where we are, so in the Iberian Peninsula, we have another layer of a situation that even leaves us in a worse position in terms of rodenticide resistance in the house mouse. So what happens here? As you all know, or not, but I'll tell you. So the Algerian mouse, the Mus pretus, occurs in the north coast of Africa and in the Iberian Peninsula up to the north of France. And what happens is that this species is naturally resistant to anticoagulants. Why? Because over evolutionary time, due to the natural occurrence of certain plants in the environment, of which Mus pretus feeds on, kind of naturally selected versions of the VCORC1 gene that provided resistance to Mus pretus. What happens is that the species barrier between Mus pretus and Mus musculus is not exactly tight, so there is not full reproductive isolation, and we still observe hybridization in the field, and that's exactly what happened. And what happened is that while hybridizing with the house mouse, the Algerian mouse moved a 20 megabase chunk of including the VCORC1 gene into the house mouse's genome, which spread well beyond the distribution of the Algerian mouse itself. So as you can see, here they call it the Spretus haplotype. That's how it was named, because it's a combination of 5 mutations occurring both in exon 1 and exon 2 of the gene that are moved in block. And so it's the yellow here, it's the yellow bit here. And as you can see, in northern Spain, it's highly prevalent, but then it was even, it reached northern Germany where there are no Algerian mice. So the hybridization occurred here, but the adaptive advantage that the house mouse got from this chunk of DNA inherited from the Algerian mouse was spreading even to up all the way to Northern Europe. Now it's way more spread. It's been detected in the UK, it's been detected in tons of places throughout Europe. So this was the framework I was sitting on when thinking about, okay, how can I contribute to this topic? So as I said before, I've been working with house mice all my life, and including during my PhD, during which I accumulated a ton of samples from lots of places, from the Azores Islands, Madeira Islands, Portugal, and continental Spain. So I proposed a PhD project to a student who's an essential. So mostly what I'm telling you guys today is mostly from her PhD thesis, and I had all this huge set of samples. Okay, let's see what's the status, what's the picture in our house mice. So basically, the original aim was to assess the levels of rodenticide resistance in a house mouse that was conferred by the VCORC1 gene. You know, the usual pipeline: sampling, DNA extraction, PCR amplification of all 3 exons, which can be a pain. Some exons more than others, but still a pain. And then sequencing, Sanger sequencing, and sequence analysis. So again, oh, this is also deformatted. Well, sorry for that. So what we were looking for, we were looking for the Spretus haplotype, so which is consistent of all these 5 mutations, and then it's called the Spretus haplotype. And then if we got just 4 of them, or 3 of them, or 2 of them, or even just 1 of them, we would consider them an incomplete haplotype. And then on exon 3, the 2 major and more broadly distributed on codon 128 and 139, as if you remember from that graph back then, were the ones that provided the highest level of resistance. So this was the original map of the distribution of these mutations in Europe. And there was a huge gap in the Iberian Peninsula, basically, back then. So I'm not expecting you guys to read this table carefully. What I just want to show is that, so these are the 9 islands of the Azores. And what I want to highlight here is that the ones that are in red, 100% of the samples that were analyzed for these islands were carried at least 1 mutation, a resistance conferring mutation. So 9 out of 12, 5 out of 21, so 4 out of 43. So these 2 islands, curiously, the biggest islands of the archipelago. But then we had São Jorge, Graciosa, and Flores, where all of the animals, 100% prevalence of mutated alleles. In Madeira, it was more or less 50-50, so 15 out of 33. In Porto Santo, again, a smaller island, it was more expressive, so 8 out of 12. And in mainland Portugal and southern Spain, again, huge numbers of resistance conferring alleles. Okay, so this is a picture for you to have a feeling of our sampling. So mostly, so basically during my PhD, I analyzed the colonization history of the islands. So that's why the sampling is more or less along the coast, because I wanted to sample areas that could have potentially been the source of animals being taken to the islands. And because southern Spain, as we all know, were also important in the maritime expansion. And there's this fight between Portugal and Spain and who got there where first. So that was the sampling size, the sampling size that I was working with. So basically, Green is the wild type. And as you can, well, I had 4. I managed to get hands on 4 animals from Deserta, and without much surprise, nobody lives there. All 4 were wild type. What was surprising was, like, again, many of the islands, all of them or most of them, were occupied by animals that carried mutations. And basically, this is one of those situations where it can only get worse, because if you naturally select the animals by using rodenticide, if you naturally select the ones that are resistant, because the other ones, the susceptibles, will die, the resistant ones are the ones who are going to breed and are going to reproduce and provide the next generation. So this can very quickly, you can get to the fixation of the mutated alleles. So a funny aspect that we noticed was that for the mutation on codon 128, so if you remember, one of those that are highly conferent of rodenticide, the pattern that it exhibits in terms of the whole archipelago of the Azores is that it occurs on the exact same islands where a clade from the mitochondrial DNA was the same. So if you see, so you have Santa Maria, Terceira, Fayal, and Flores, and then here you have clade F, which basically is a clade, a mitochondrial DNA clade, who originates from Northern Europe, so Norway, Iceland, Northern Scotland, Northern Germany. Basically, those islands where this mitochondrial clade occurred are the exact same islands where codon 100, the mutation on codon 128 also occurs. So this hints me that there's obviously a huge impact on the colonization history of an island, because of course animals had to get there indirectly, transported by humans. There's no way a mouse could swim to the Azores, so they had to be transported. So if the place where they originate already carry these mutations, you take them with the mice and then they can be selected. If they never reached the island, so if that clade F didn't reach São Miguel, so maybe never there was very few contributions, very low contributions of mutation on mutations, resistance conferring mutations here, because the mutations didn't get there and there was no time for in situ emergence. Okay? So to wrap up this bit, clearly what we can see here is that smaller islands show a significant presence of the homozygous version of the mutation on the 139 codon, and that this mutation is completely widespread throughout the Portuguese archipelagos. 100% of the analyzed animals of 3 of the Azorian islands carried at least one resistance conferring mutation. The Spretus variant is the most common in mainland and southern Spain, so not surprisingly at all. Mutations in the VCORC1 gene seem to be present as natural variation among rodent populations and are positively selected when in strong contact with rodenticides. Just one parenthesis that I didn't mention. Carrying these resistance conferring mutations comes at a cost, okay? The cost benefit is obviously favorable when you don't die, because if you have to eat rodenticide, or if you are in contact with rodenticide, it's obviously much better to pay whatever price than dying. But there is a price. So there is a price to carrying these. So the number of offsprings is lower. So the animals have a lower fitness when they carry these mutations. So it really needs to pay off to maintain these alleles in the population. It pays off if you are in an environment that is rich in anticoagulant rodenticide. So finally, mostly I have like remote adding to this, because obviously in remote island systems, the population is way more closed. It's a closed system. There's not huge contributions of incomers from mainland. So the colonization history, the original colonization history, seems to be really, really important in determining the patterns of rodenticide resistance. So, okay, so this is the story on the house mouse. So what about rats? Okay, so I had a project a few years ago, and we called it Trojan rats. And basically, our sampling sites were the city of Lisbon and the city of Ponta Delgada, two port cities. This is a map of the maritime traffic at any point in time. And so I was already interested in this time on these questions, and I wanted to understand if rats are still to these days being transported all over the world, if two cities that are highly connected in terms of maritime traffic would somehow share or exchange rodenticide resistance alleles. So again, we sampled in both cities, Lisbon and Ponta Delgada, in a 10-kilometer radius from the port. So both cities have more or less the same shape. So basically they are like this, the port is here. So it was like a perfect 10-kilometer radius. And we got loads of rats. Praise to the crew who worked in this project, because it's not everybody's cup of tea to work with wild rats. So we analyzed 203 tissue samples from both black rats and brown rats. Again, the same pipeline, DNA extraction, PCR amplification. Here we focused exclusively on the exon 3, because from the literature, we realized that the effort, both in time and money, particularly in money, didn't pay off to analyze the first and second exons if we didn't find anything on the third one, because the third one in black rats, there's obviously not the case with this predation haplotype, because of course they don't interbreed. So that was a particular aspect of the house mouse that we didn't get here. So we focused on exon 3, and then the PCR project was Sanger sequenced, and the sequences were analyzed to look for mutations, and we found none. Wah, 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 wah. It was like one of those moments, like come on, all over Europe, there's tons of mutations in rats, and we found 0? Like, seriously, what's wrong with us? Come on. Well, it's a good news for pest controllers, we would expect, because of course there's no problems about, okay, so which rodenticide should I choose? Because certain rodenticides are clearly out of hand if you have certain mutations, because you saw on that graph in the beginning, it's pointless to use bromidilone if you have the predation haplotype for house mice, for example. So in rats, in this case, so if they are genetically susceptible to anticoagulant rodenticides, then pest control should be effective, okay? They are susceptible, they should die. And the exposure to non-target wildlife should be minimized, because the animals eat, the animals die, end of story, okay? Let's see if that's the case. So this was kind of a side project because that was not originally planned to run this task and this part of the work, because it never crossed our minds that we wouldn't find any mutations on the rats. So we partnered with a group in Gran Canaria, and apparently nobody in Portugal quantifies anticoagulant rodenticides, and after a year or almost a year lost in attempts, okay, let's talk with who knows. And so we partnered up with them, with Certox, and that was like the best decision ever. So basically, we had liver samples kept from all the animals, originally from the live trapping that we did in the project. We took 1 gram of liver, homogenized, and then, of course, lots of intermediate steps, and we quantified all the FGARs and all the scars simultaneously in the same run using LC-MS, so liquid chromatography coupled to mass spectrometry. So, and we found no FGARs whatsoever, okay? We only found second generation anticoagulant rodenticides, which on its own is quite worrying. So basically, what we realized was that 81.4% of the animals that were analyzed bioaccumulated at least 1 scar in their liver. So as you can see, 18.6 had none, and this is like across the sampling. So juveniles, adults, all sorts of animals. So we really wanted to see if that was really across the exposure of the animal, all animals. We didn't select for specific age groups. So 18.6% we detected nothing. In clear almost 34 we detected 1, 34.5 2, 12.4% of the animals had 3 different rodenticides accumulated, and 0.6% had 4. So they were a walking packet of rodenticide, basically. As you can see, all animals together, not divided by species. They accumulated, so this is yellow is Lisbon, green is Ponta Delgada. Clearly, bromidilone and Brodifacoum are the ones that lead the list. Okay, so the most commonly found, and also we know the most commonly sold rodenticides. Lower expression of difenacoum, difethialone, and flucomafan, but all 5 of them were detected. We also tried to see if there was a species-specific pattern. There wasn't. So black rats and brown rats didn't have any significant differences in the quantification of rodenticide, and also there were no differences between cities. So this dashed line here at the 100 nanograms per gram is supposed to highlight the value above which animals show toxicosis. What does that mean? It means that the animal is exhibiting a physiological response, a clinical response to the rodenticide. So basically, in English/Portuguese, it's getting sick, okay? And as you can see, there's a lot of animals, not so much in black rats from Ponta Delgada. They were all below this threshold, but there might be a reasonable explanation for this, is that on the contrary to Lisbon, where black rats occur exactly where brown rats occur in the city, in Ponta Delgada, you have a much more rural city. So these guys were found more in less urbanized areas. So little patches of forest, that's where we most got black rats. So brown rats were most caught in the urban matrix, and brown rats more in the agricultural and foresty bits. Still, they accumulated tons. So that was the sum of all rodenticides. Now partitioning by biocides, so by individual compounds, we have, again, Brodifacoum and bromidilone, like these two first plots, being the most common. The curiosity here is that in Ponta Delgada, and please keep that in mind, actually, the one that we found with the highest mean was diphthiolone, which is like hell on earth in terms of toxicity. It's the worst. It was the last one to be developed, the last rodenticide to be developed, and it's really huge. As you can see, this one and flucomophen, they were barely found in Lisbon, but in Ponta Delgada, really a lot. I'll come back to that later. So besides the impact that this has on the rodent control and the management of rodent populations, there's another thing we have to think about. So it's the biomagnification route that takes place when we are putting these things in the environment, okay? So it's not just rats that eat the rodenticide, okay? We aim for that, but that's definitely not the case. There's other non-target species who are directly feeding on the pellets. Insects, slugs. Slugs are, we were just walking in the forest in your experimental plot a while ago, and there was like a huge slug, and we were actually talking about this. So slugs can literally munch on a pellet of rodenticide, even turn blue if that's the case, and nothing happens to it. So if a blackbird or hedgehog or whatever comes and eats it, there you go, like huge intake of rodenticide. And then you have, like I said, the biomagnification route. You have passerine birds feeding on insects and slugs. You have rats and mice themselves eating them. And then you have the top predators, and then you have the secondary exposure. So these guys never have to touch or smell a pellet to be exposed to huge quantities of rodenticide. In the UK, actually, the law has recently changed because, you know, maybe the biggest community of bird watchers lives in the UK, and they love birds, and they make everything they can for birds in particular. They're very into this group. And there's a huge campaign trying, so this is a huge outdoor. So rat poison kills more than rats, and don't poison my dinner, and please stop poisoning our food. So recently it was even forbidden by law for normal people like you and I to actually go on a store and buy second-generation anticoagulant rodenticides. So huge that the problem is there. Okay, you cannot do it. You need to be a pest controller to apply it. You have to be certificated to that. It's impossible for someone going to Leroy Merlin or IKEA, not IKEA, sorry, Aki, and buy, or Continental or wherever you go, because you can find rodenticide anywhere on these over-the-counter options. So, okay, let's see what's happening to the top predators then. Because we already knew that the target species, the target rats that in theory should have been dead with huge amounts of rodenticide in their liver, they weren't. They were still walking around because they were killed, because we performed the necropsy. So they were alive until we performed the euthanasia. So they were fine. They were absolutely active animals in the ecosystem. They were doing their thing, okay? So they were available to be preyed on, okay? So what did we do? We contacted wildlife recovery centers and asked for liver samples from the species of birds of prey that had entered the center and died there. Exactly the same protocol, 1 gram of liver. So we analyzed 211 birds. The same process, aiming to quantify, simultaneously quantify all FGARs and all scars. So these were the species that we managed to get really interesting numbers of. Ethinoctua, butiubutiu, titualba, bububu, falctinunculus, and strix alux. So basically, we have representatives of daily animals, of nocturnal animals, little animals like falctinunculus, so the common kestrel is a pretty small bird, and bubububu is a huge one. So we had basically some of them preyed specifically on small mammals, others were more generalistic. So we wanted to take a look at that. And of course, these were available in relatively large numbers. So unfortunately, I won't be able to show you data from the north of Portugal because we didn't manage to get any access to samples from north of Castelo Branco. So we got samples from Crassa, which is here in Vila Nova de Santo Why don't I ever remember the name of this one? Ceres, sorry, which is from Castelo Branco. Rias, here in Olhão. And we got also a few animals from Madeira Island, provided by the IFCN. Here you can already see the distribution of the 5. So again, we found the same 5 scars, no FGARs, yes, again. And this is the scary bit. So there's also a few animals that we ran anyway because we had the samples, but because the numbers were so low, so 4 and 5 and 7 milvus, still 100% of those 4 had rodenticide, 100% of these 5 as well. So basically, the lowest percentage of detection was found in butiubutiu, so the common buzzard, with 64.7% of detection rate. So 4 of the species, Titualba, Strixalug, Falctinunculus, and Bubububu, had at least 80% detection rate of rodenticides. So here, instead of showing you the 100 nanograms per gram threshold that was considered for rodents, I am showing you 2 other thresholds, the 80 nanograms per gram and the 40 nanograms per gram. Why? Because very recently, literally 2024, authors have realized that the birds of prey are even more sensitive to the rodenticide than the rodents themselves. So the toxicosis threshold, so the concentration above which there are signs of illness related to the intoxication with these products, for Titunidae, so for the family where Titualba is included, is actually 40 nanograms per gram. For the other ones, 80, okay? which is pretty scary, is that, for example, for Boo 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 Boo, almost all of the box is inside these two thresholds. And it's not that they were very little, that the sample size was very little. There were 22. Animals. Falctinunculus also huge median values of rodenticides, and so T2, Bubu, and Falco being the ones that clearly show the most concerning results. So here is an analysis by family. So apparently there is no significant difference between the family the animals belong to. So it's pretty much whatever. You are a bird of prey, you are affected by rodenticide. And again, the distribution of the different rodenticides also follows the same pattern. Again, Brodifacoum and Bromeliad being the clear first and second most detected rodenticides. Another view of the data now, a geographical view. So all birds together, divided or separated by district where the animal was found. So Santarém, if you most likely know, it's a region of the country where tons of agriculture, tons of the produce, the vegetables that we eat every day come from. It's a highly rural area where there's lots of agricultural production. And the birds there are really under a huge level of exposure. Again, like you can see here also, Bromeliad and Brodifacoum, except for Madeira. If you remember, the rats data in the Azores, they were the only ones that had huge amounts of diphethylone, okay? So there's a bit of a unfinished puzzle here. So we have rats from the Azores and we have birds from Madeira, okay? What do they have in common? They are islands. What do islands have in common with each other in terms of rodent populations? They have loads of them, okay? So oceanic islands are very poor in mammals, all of them or most of them being invasive, okay? And in the case of Madeira and the Azores archipelagos, these are the only rodents inhabiting the islands. So house mouse, black rats, and brown rats. And virtually, well, I'm not going to say virtually no predators, but also a very limited number of predators. There's a couple of birds of prey in Madeira and in the Azores. The terrestrial carnivores are also very limited, mostly limited to cats and dogs. So there's not the level of diversity so there's no foxes, there's no geneta, there's no herpestes, so there's nothing that usually eats rodents on mainland. So the populations on the islands just explode. And this is why if you want to open a rodenticide, not a rodenticide, a pest control company, the place to be is the islands, because there's like one at each corner. They have so many because it's a huge problem. Rodents, pest rodents are a huge problem on the islands. And clearly, even though we don't have data on the bioaccumulation of mice or rats in Madeira, we can for sure say that they must be full of diphethylone, okay? Again, here, the 80 and the 40 nanograms per gram thresholds are highlighted. And for many of the rodenticides, many animals laid exactly on that huge interval. So what we can conclude from this is that raptor species are highly affected by anticoagulant rodenticides, and none of the studied species can be considered safe or kind of getting away with it. It's not happening. Both diurnal and nocturnal species were highly contaminated with scars. Bromeliolone and Brodifacoum were the most commonly detected rodenticides on mainland, but diphtheolone was the most prevalent in Madeira. And actually, this was something that last night, I just realized this last night, and I was checking. So because this manuscript is like, we are literally a few days away from submitting. And there was a sentence there about one of the species, and I said, okay, wait, let me check for the other ones. So if you remember, Bububu, Falcotinunculus, and Titualba were the ones landing on the top, the boxes, the whiskers and the boxes the box plots were landing on that 40 to 80 nanograms per gram interval. And I actually went to check last night. So Bububu, Falcotinunculus, and Titualba used to be in the last red book, least concerned. Bububu is now near threatened. Falcotinunculus is now vulnerable, and Titualba is now near threatened. It can be a coincidence, and it can be that these species are facing other challenges. But if I had to guess, I would say that for sure this isn't helping. Okay, so this aspect, this toxicological aspect, is not helping, because all vertebrates that become in contact with anticoagulant rodenticides will suffer. We die if we eat rat poison in a sufficient quantity. So this is clearly something that needs to be monitored and looked at and thought about. You may say, and of course, I'll give you that, this sampling is very biased, okay? It's pretty obvious because our data comes from animals that were either found dead already or died in the Wildlife Recovery Center where they were taken, most likely because they were found injured or unable to fly or whatever. So we're totally aware that this is a biased data set. So these animals were already ill or ill enough to be hit by a car, run into an electric wires, thank you, run into a window or whatever that made them, that led them to be taken to a wildlife recovery center. Still, until they were dead, they were exhibiting these levels of rodenticides in their liver. So that's not a good, it's not something that we think, oh, okay, there are tons of other birds that must be fine. Well, of course, but if these animals died as a direct or indirect consequence of the exposure to the rodenticides, it's of course an animal that will no longer reproduce, that will make the population weaker into the future. That was something I wanted to say here. And well, so some just like trying to zoom out a bit and look at the problem from a bit above. Well, food for thought, not that I want to feed you any rodenticide, but just a few numbers and an illustration of what's the situation. This is the Azores, this is the São Miguel Island. So as you can see here here, 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 here. See, different. This is little bags of pink rodenticide. This is like a blue pellet. Also here, also here. This is illegal, by the way. You cannot dispose rodenticide like open air. However, the rodent problem is so gigantic that the farmers just don't care, or they just don't know, because they just throw it like corn to a pigeon. So this is just everywhere you go, you find this, and they are not even trying to hide it, because these should be inside a bait box, stuck with a wire, unable to leave the box. So the animals have to feed inside the box. So they cannot drag it. Other animals cannot drag it out. And basically, this is available for anyone who wants to eat it. Okay? So there was a report from the regional secretary of Agriculture and Forestry in the Azores that in the period between 2009, that's a bit dated already, but I don't have any more recent data. Between 2009 and 2019, the regional government acquired 773 tons of rodenticide. Guess which? Which rodenticide did I buy? It's the Azores. Diphtheolone. Yeah, and they distributed it throughout the 9 islands, which total at 2,300 square kilometers in area at economic cost of over 1.6 million euros. So, and this is just what the government bought. This is not what the pest controllers applied. This is not what people bought over the counter. So they bought it, and then whoever wants to goes to the junta de freguesia, I don't know, how do you call it? Like the little parishes, and say, oh, okay, can I take 2 kilos? Can I take? And then they, oh, okay, go ahead, sir. And then they go like this, okay? So this is not considering what people bought, what the pest controllers used. That's just what the government bought, okay? So definitely this number is clearly underestimated. For scale, 773 tons of rodenticide looks something like 155 African elephants. And 2,300 square kilometers is an area the size of Luxembourg, which we can cross in half an hour by car. That's a lot of rodenticide in such small area. So this is, in the Azores, what we are dealing with. Considering that we already know that the mice, which is like half of the problem, so they're still the rats, the mice are virtually all resistant to it. So basically, they are paying to feed them, okay? Because the pellets, they are really tasty. They make them really, really palatable, because otherwise the animals wouldn't eat it. So the percentage of the active ingredient in the pellet is like 0.0025%. It's like ridiculously low. So they are literally feeding them, okay? So are we hostage? Are we held hostage of anticoagulant rodenticides? Well, kind of, because the alternatives are not real alternatives. So yes, there's non-anticoagulant rodenticides. In Australia, they use zinc phosphide, which has an acute mechanism of action. Literally, the animal eats that, it sublimes as it eats, so produces a gas, and the animal is dead within minutes. But in Australia, they are dealing with mouse plagues with millions, literally millions. This is not like an overreaction, millions of animals per hectare. If you Google mouse plague in Australia, you will be looking at that with your eyes popping off your head. Because it's like they go over people in hospital beds, when they go into everywhere, they crawl into people's beds. It's just ridiculous. It's something out of a horror movie. There's another option called calciferol. So this is the big word for vitamin D rich rodenticide. It's also not great because it's the death is probably even worse than the one with the anticoagulant rodenticide, where the animal dies out during days. That's why you don't find the rats or the mice near the place where you left the rodenticide, because they literally start to feel sick and they go to their nest and die there. That's why you most likely won't see them. The calciferol doesn't work in terms of anticoagulants, the way anticoagulants do, but they calcify your soft tissues, including your heart muscle. So basically, the animals die of heart attacks, and their kidneys get overloaded because the soft tissues and even the endothelium of the capillaries gets thicker, because there's calcification of these tissues, of these soft tissues, and it also takes a long time to die. So it's also not, on an animal welfare perspective, it's also not an alternative. Well, integrated pest management, yeah, right. That's very beautiful to say, and all the pest controllers say they do it, but come on. When we are dealing with the amount of animals that we are, for example, dealing on the islands, there's no way that just combining techniques of sanitation, keeping everything clean. How do you keep an agricultural area clean? How do you keep a cow production, a dairy farm? How do you keep it clean? Okay, so the animals don't go there. Yeah, structural exclusion. Yeah, and then they find a hole this big, and if the head goes through, the whole body will as well. So in theory, it may help if you have low levels of animals. If you have a pest size population to control, that won't work. So gene drive, that for sure would solve the problem, for sure. I found this cartoon, like, oh, I might know how to address the situation. They're crawling over her. And yes, in theory, I know there's people working on this, on the CRISPR-Cas9 technique, gene editing. So on Mendelian sort of inheritance type of the way genes pass on to the progeny, you have a 50% chance of passing a certain gene. If you edit it, you can make sure that all your progeny ends up with a gene that will be incompatible with life, or you can just make males and the population will just collapse on its own. In theory, yes. And on islands, it would be for sure amazing. But there's always a but. And the but here is that there's a ton of ethical issues. And just to mention a couple, well, how do you make sure that your edited animals will not escape from your island? How can you make sure that your edited animals will not mate with other animals from a different species, like the mus musculus mus pretus situation, and jump the edited gene to another species, and then we will end up with a collapse of a population or a species that we didn't want to affect. So yes, in theory, it looks pretty much what would do it. But the ethical issues around it are so many that I am not sure if that will ever see the light of day. So it's Friday afternoon, and I don't want to bore you too much longer, and my time is about to be up, right? Yep. So basically, just a few take-home messages. The mission of managing pest rodent populations remains, and there is apparently no quick fix on site. Anticoagulant rodenticides are all over the world, are completely ubiquitous, and no valid alternatives are expected to rise in the near future. Bromidilone and brodifacoum, two highly toxic and persistent scars, are the most widespread rodenticides detected in both target rats and non-target birds of prey. Something that I now just remembered what I wanted to say before is that one of the reasons we might not have detected scars, well, two reasons actually. So the first one is because nobody bothers to use them anymore, because they know that how what's the word? They don't work anymore, useless. Exactly, thank you, Guy. Exactly, because it's pointless for you to invest in a rodenticide and you just have the feeling that the animals are only feeding on it. And another reason is that because they are not as persistent, so they are eliminated by the animal and they may be more efficient at getting rid of it and metabolizing and getting rid of it through the feces. And their persistence is much, much larger, up to months for the scars in comparison with the scars that may only last a few days in the body. So rodenticide resistance only adds another layer of challenge into this already huge horror movie, because if you have a population, a rodent population that is resistant, it's expected, that's not the case. So the population that we analyzed was susceptible and still they were accumulating loads. Unfortunately, I didn't have liver left from I did, but it's in ethanol and it cannot be used, from the house mice in the Azores. So that would be brilliant to see, okay, if susceptible animals accumulate this much, resistant animals, they will accumulate even much more expectedly. And of course, that will make the exposure to wildlife even more problematic, because a single owl may feed on 3 or 4 mice every night. And if they feed on 4 resistant mice, only in that night she will upload a huge amount of rodenticide. Yeah, again, that's what I was saying. Even genetically susceptible rodents can bioaccumulate high levels of rodenticide, sometimes well above the toxicosis threshold, and alternative mechanisms to VCOR-C1 mediated resistance must be playing a role here, because this was clearly unexpected, that we found such high amounts of rodenticide in animals that were supposedly that they should be dead. So there's a lot of work to be done. I'm hoping to contribute a bit further with this. So we are just about to start a project on the impact of the microbiome on the, so if the animals themselves, their own microbiome is helping by producing enough vitamin K, which is the antidote to this. If you have a dog or a cat that eats a rodenticide pellet, basically you can take it quickly to the vet and the antidote will be a shot of vitamin K. So the recycling of the vitamin K is interrupted because of the rodenticide, but if you just get a shot of vitamin K, the animal may have a chance to survive. So I'm trying to explore this avenue and try to see if the animal's microbiome is actually helping with the production of enough vitamin K to keep them at least going on for a while. So the acknowledgements and a center, so that's my PhD student. So like I said in the beginning, this work, it started a long time ago. So the collection of the samples started in my own PhD on another life. But the actual work, the VCORC1 work, the quantification is all in her thesis. So thanks to all her hard work so far. And so Beatriz Martin Cruz and Otávio Luzardo are the colleagues from the Gran Canaria Sertox lab, which have been absolutely priceless to have as collaborators to run the rodenticide analysis. And my longtime mentor, Professor Maria de Luz, of course, our small lab, the whole gang, so 3 representatives here today. Thank you, guys. And the Trojan Rats crew, who also were great troopers and had no problems of dealing with huge 800 grams rats. So, and of course, thank you all for listening. And I will take any questions you may have for me. Thank you.